Welcome everyone. I'm Dr. Chitra Devi, Assistant Professor of the Botany Department, Psgr Krishnammal College for Women, Coimbatore, and I'm very eager to take you into the third part of the session on plant tissue culture. In the first two parts, we saw about the history, the basics in designing and organizing a plant tissue culture laboratory, the techniques involved in plant tissue culture with a few of my research examples, and in this session, we shall deal with the applications of plant tissue culture. Plant tissue culture has broad applications in several areas. These applications are generally classified into three categories: basic research, environmental issues, and commercial applications. Basic application covers the concepts in understanding the physiology, considering the molecular pathways in cells, whereas environmental application covers conservation strategies to preserve the elite germplasms for a longer time. Current research in plant tissue culture is highly focused on commercial applications such as crop improvement, secondary metabolite production, and various strategies for inducing genetic interference. Orchidaceae is the largest family with an estimated 25,000 species placed in about 880 genera and with 80,000 hybrids comprising 10% of the angiosperms. They are most valuable as potted crop around the world. In India, orchids form 9% of the flora. It is estimated that about 1,300 species belonging to 140 genera are present in the Himalayas, with others scattered in eastern and western Ghats. The Western Ghats, one among the 34 hotspots of biodiversity, consists of a total of 310 orchid taxa, of which 118 are endemic to that region, and about 190 taxa are also present in other parts of India and the world, according to Camp 2001. Orchids feature predominantly in many national red data books, and the abundance of many orchid species is believed to have fallen to critical levels in recent decades. The causes may be attributed to habitat loss, loss of pollinators, absence of specific fungal symbiont, indiscriminate collection from the wild, etc. Orchid seeds are produced in large numbers but are exceedingly small. The endosperm development stops at an early stage, and in many cases, definite endosperm tissues are not apparent at seed maturity. In addition, the embryos are highly reduced. The combination of small seed size and fungal symbiont requirement largely precludes the propagation via conventional seed propagation protocols. Accordingly, most of the research on the development of orchid seed germination protocols focused on symbiotic in vitro germination. Successful protocols were developed for several species. However, the process was complicated by the diversity of fungal symbionts required across orchid taxa and the difficulty in isolating and culturing these fungi. In the 1920s, the process of orchid seed propagation was revolutionized by Louis Knudsen with the demonstration that nutrient media could be used as a substitute for symbiotic in vitro germination. In this example, we shall see the micropropagation of Coelogyn prolifera, an endangered epiphyte distributed in the hills of Sikkim and Meghalaya at an elevation of 800 meter, 2,300 meters, and Vanda testacea, an epiphyte usually on ficus species and distributed in the Himalayas, Western Ghats, and in Andaman and Nicobar Islands. We have used two basal media, Murashige and Skoogs and Knudsen C, along with 3% sucrose and agar. Let us see the results we have obtained. We have used only MS basal medium and Knudsen C basal medium. So here you can see Coelogene prolifera's response on MS basal medium. Almost all the seeds have germinated and they have formed pseudorhizoids. And the seeds of Vanda testacea on MS basal medium, most of them have died. These brown spots indicate the seedlings which have died because of nutrient deficiency in the medium. Here you can see the germination of seeds of both the orchids, Coelogene prolifera and Vanda on Knudsen C basal medium. Here you can see Coelogene prolifera. Most of the seeds were not able to develop into maturity on KC medium. However, all the seeds of Vanda are maturing on KC medium. So from the study we can infer that MS basal medium was suitable for Coelogene prolifera and KC basal medium was suitable for Vanda testacea. Now let's see the next example. Here in this slide you can see the stages in seed germination of orchids. So this is the seed which is covered by a seed coat, and you can see the embryo. So once the embryo is maturing, the seed coat ruptures off and then the protocorm is formed. So protocorm is the primary organ formed during seed germination. So in this protocorm, all the food materials are stored. So this protocorm again starts to grow and produces pseudorhizoids. These pseudorhizoids, they are not true roots. They are helpful only in absorbing the nutrients from the medium. So once they absorb the nutrients, the shoot apex starts to develop, and the shoot apex forms the first true leaf. And then you can see when the first true leaf starts to emerge, the pseudorhizoids are all gone and you have the first true root initial. So these are all the complete stages of seed germination in orchid, asymbiotic seed germination in orchid. Here we can see another example of Coelogene stricta. This is also an endemic plant of the Western Ghats. So here we have used MS basal medium with 3% sucrose and with or without hormones. So we have used just basal medium, and also we have used basal medium supplemented with either BA or IAA or BA plus IAA in all these concentrations. So you can see the stages in seed germination. So picture A, seeds. Picture B, you can see all the seeds have germinated on MS basal medium, and all the germinated seeds have produced pseudorhizoids, and all the seeds forming pseudorhizoids, they have developed the first true leaf initial. And you can see the leaf initials in close up, and the pseudorhizoids also you can see here like spider webs, and here you can see the first true leaves. So in this cluster you have to separate each individual plantlet and grow it separately on a medium. So here you can see the bulged area, the protocorm, the root initial, the actual root, the first leaf and the second leaf. And when this plant matures, you can see it produces two types of roots, and also the shoot starts to develop. So once it attains a specific maturity, it has to undergo the phase of hardening and acclimatization. Now here we have studied another method, MS medium with IAA alone and in combination with BAP. So here you can see the results. The dark spots indicate the death of the germinated seed, and also the plants do not completely form first leaf, and even if they form, the leaves are of a deformed nature. So we find that hormones have a definite effect on the germination of seeds of orchids, whereas medium without hormones will give you normal, healthy plantlets without any abnormalities. Synthetic seed preparation is another application of plant tissue culture. So we can see an example of encapsulation of in vitro nodal explants of an important medicinal plant belonging to Asteraceae in the next slide. So first let me explain what are synthetic seeds. Synthetic seeds are also called as artificial seeds. They are defined as a somatic embryo or any other vegetative propagule which has an artificial biodegradable coating entrapped in a nutrient medium supplying carbon sources, mineral nutrients, vitamins, and growth regulators. So, what are the uses of synthetic seeds? Multiplication of non seed producing plants, ornamental hybrids propagated by cutting, or the propagation of polyploid plants with elite traits, propagation of male or female steroid plants for hybrid seed production. Cryopreserved artificial seeds may also be used for germplasm preservation, particularly in recalcitrant species such as mango, cocoa, and coconut. All these seeds will not undergo desiccation. Transgenic plants, which require separate growth facilities to maintain original genotypes, may also be preserved using somatic embryos. The only technology realistically amenable to the extensive scale up required for the commercial production of some clones. So, synthetic seeds are of immense value. So, as I have told you previously, this is an example of an important medicinal plant belonging to Asteraceae. Here, we already produced multiple shoots from the node of that plant, and these shoots we excised and we took the nodes from these shoots and encapsulated them. So the in vitro nodal explants obtained from multiple shoots on MS basal medium at pH 5.6 supplemented with 0.88 molar sucrose, 0.8% agar and BA were used for the encapsulation experiments. How did we prepare the encapsulation matrix? Sodium alginate plus calcium chloride matrix was used with or without growth regulators such as BA, kinetin, individually or combined with IAA and autoclaved for about 20 minutes for sterilization. So, we conducted this experiment in two ways. First, we incorporated the hormones in the medium. Next, we incorporated the hormones in the matrix. So, hormones, when incorporated in the medium, gave us such results. So, you can see the node, only the node starts to develop into a shoot initial and a root initial. In kinetin, it forms a callus and only one shoot is developing. So when IAA and BA are used in combination, you can see something happens here which is in no way related to multiple shoot induction. Here IAA and kinetin when added in this combination, you can see some 2 or 3 shoots being produced from the nodal explant, encapsulated nodal explant. So IAA and BA at these concentrations have given rise to multiple shoots. Here, the hormones were incorporated in the encapsulation matrix. The medium was just basal medium. The encapsulation matrix contained the hormone BA. And you can see just a shoot and a root initial has formed. That is, the encapsulated nodal explant has produced a complete plantlet. Here you can see the basal medium was not MS but Schenck and Hildebrand medium. So here you can see numerous shoots have developed and beautiful leaves have developed, and you can see the roots, they have developed enormously. And then here you can see on MS basal medium, IAA in combination with kinetin produced a different response, a response different than both these. So when the plantlets are about 2 to 3 centimeters, we cut the plantlets and go in for rooting. That is because most of the roots produced most of the roots produced in this medium are saprobic in nature. That is, they only perform the function of absorbing the food materials already available in the medium. So, such roots, when you take it to the hardening process, will not help in the survival of the plant. Therefore, we cut the shoot and we go for rooting again, irrespective of whether the shoot has developed the root already. So once the roots are completely developed, we go in for hardening. And then after the hardening under laboratory conditions, we take out the plant to external conditions, and after this they go into the greenhouse. So with this, we have come to the end of this session. Dear friends, the applications of plant tissue culture are a vast ocean of a topic, of which I have been able only to cover a few examples. I sincerely hope that through this around 40 minutes journey, you have gained something useful. I would like to thank my head of the department and all my faculty friends for having seen the enthusiast in me. I'm grateful to the management for giving us such a solid platform to showcase our research strength, and I sincerely acknowledge the contributions of my students whose research work I have quoted as examples. Thank you.